Cells such as this fibroblast form the building blocks of tissues, organs, and entire organisms. As such, an understanding of how they respond to various stimuli is vital to the study of both health and disease. In order to gain this understanding, it is necessary to first simplify the picture. The stimuli that affect the behavior of cells come in a variety of forms. These include chemical stimuli, both externally derived, such as caffeine, or internally, like neurotransmitters or hormones. Then there are electrochemical stimuli, such as those that propagate messages along neurons. Cells are also able to respond to physical stimuli, such as fluid shear stress in blood vessels, or the stiffness of the environment surrounding a cell. It is the last of these stimuli to which we have directed our research. The first step is to understand how a cell attaches to its surroundings, or to a surface in a simplified model. Looking at a cell from the side, we see that it interacts with the surface through a series of discrete attachment points, or adhesions. Let's look closer at one of these adhesions. Here we see the lipid bilayer that is the basis of the cell membrane. Passing through the membrane is a protein called integrin. Outside of the cell, this protein binds to the extracellular matrix that surrounds the cell, represented here as a dense mesh. Inside the cell, the question turns to what else binds to integrins that may give the cell the ability to sense physical stimuli associated with the extracellular matrix. This is the protein Talin that binds to integrins inside of cells. Our previous research has shown that the structure of this protein acts like a spring, i.e., when it is exposed to tension, it extends and unfolds. One end of the Talin protein binds to integrins on the inside of cells. This connects it, through the membrane, to the extracellular matrix. At the opposite end, it binds to the actin cytoskeleton of the cells. This cytoskeleton acts for the cells in much the same way as the skeleton of a human body, providing structural support. And similar to the muscles that attach to a human skeleton, the cell's cytoskeleton has contractile elements that can generate force. Talin is one of only a handful of proteins that connects both to integrins and to the cytoskeleton, forming a bridge between the two. The contractile nature of this cytoskeleton means that this bridging position results in tension being applied to the Talin structure. How does this help the cell sense its external environment? It is helpful here to equate the extracellular environment to another spring. This reduces the problem to that of springs in series. The Talin spring is connected via a rigid link to the spring representing the extracellular matrix. Tension is then applied through the force-generating units of the cytoskeleton. If the ECM spring is soft, it will be extended, while the Talin spring will remain at its original length. Conversely, if the ECM spring is stiff, it will remain compact, and the Talin spring will be extended. In cells, this extension can be up to several times Talin's original length. Looking at our whole cell from the side, we can now replace our hard surface with the dense mesh of the extracellular matrix, which is attached to a number of integrin molecules, forming the discrete attachment points. Inside the cell, these attachment points are connected to a number of springs, the Talin molecules. These in turn are connected to the cytoskeleton, providing multiple sites at which the sensing of physical stimuli can take place. The question then turns to how Talin translates extension into the biochemical signals that cells are governed by. In our research, we focused on a small region of the Talin protein, the R7 to R8 subdomain. This subdomain can be equated to a small section of the Talin spring. This particular part of the spring binds to a molecule called DLC-1. Crucially, DLC-1 binds to this subdomain when it is tightly coiled. If Talin, particularly R7 to R8, is extended under tension, DLC-1 can no longer bind, and the loss of this interaction has an effect on a variety of cell signaling pathways. Here we see a structural representation of the binding of DLC-1 to Talin and its subsequent unbinding under force. To study this mechanism, we introduced a molecular clamp into the R7 to R8 subdomain, such that even when Talin is extended, the structure that binds to DLC-1 is preserved. This resulted in changes to cell behavior, such as force generation, which we measured by seeing how a cell pulls on elastic micropillars, as shown here. But why is this research important? The ability of a cell to both apply force and sense physical stimuli from its surroundings through these discrete attachment points has wide-ranging implications. During embryogenesis, cells shape tissues by sensing and responding to the geometrical and physical properties of their environment, governing the differentiation of stem cells and the shape of individual tissue structures. This morphogenic process has been shown to be governed by a protein called Rho-A, which itself is controlled by the interaction of DLC-1 with Talin. Fibroblast cells control wound healing through a tightly controlled cycle of activation and deactivation. This cycle is partly governed by the ability to sense and create forces, both of which are once again controlled by Rho-A downstream of Talin's interaction with DLC-1. Poor regulation of this cycle can lead to fibrosis and conditions favourable to cancer development. Looking at cancer, tumour cells rely on migration to invade and colonise other tissues. This cell migration is controlled by DLC-1's interaction with Talin, as demonstrated in this video that formed part of our research. It is instructive at this point that DLC-1 is an acronym for deleted in liver cancer 1, as the loss of this protein in liver cancer and a variety of others has been found to lead to a dysregulation of physical stimuli and enhances cancer progression. If you are interested in learning more about these processes and their implications, you can find our research here. This was a joint project between Imperial College London and the University of Tampere, and if you'd like to discuss our research, please be in touch.